So welcome to this case study where we want to integrate ReadStore with Scanpy for single-cell RNA-Seq analysis. So ReadStore is a data repository for omics data, including raw, processed, and metadata, and it helps you to build a single source of truth for all the data sets that you need for bioinformatics analysis. And we want to demonstrate here how you can use it with Scanpy. So Scanpy is a fantastic tool for single-cell RNA-Seq analysis in Python that is widely used in the community. And we want to see how we can even boost our analysis workflows using a combination of ReadStore and Scanpy to facilitate import of datasets and annotation of metadata. The dataset that we want to look at here comes from a publication by Sylvain et al. In Cell Reports. And the authors looked here at a condition that is called SAVI, sting-associated vasculopathy, with the onset in infancy. This is an autoimmune disease that is starting in child age, and what the authors did here is single-cell RNA-Seq processing of blood from patients that suffer from this condition and control donors. We have a total of 17 data sets that come from different treatment regimen and a set of clinical and experimental variables that need to be managed in order to make sense of these data. And this we want to do using ReadStore. So what we will see now is that we're going to import those data sets from our ReadStore database using the Python SDK. We're going to also load the metadata from our ReadStore database for each of those data sets. We're then performing a quick filtering of these data and upload the filtered data set again to our ReadStore database so that we have them available for future analysis. And finally, we will run through quick single-cell analysis where we make use of these metadata to investigate differences in the single-cell profiles. So let's start in the projects overview for our ReadStore app. And here we can see that we created a project that is called SAVI study that will hold all the data sets for our analysis, and we can later then access the data sets via this project. We can go to the detail view and see that we have some metadata attached to this project. Those are identical for all the datasets that are associated here. And if we move to the datasets page, we can again filter for this project and we get a list of all the datasets that are attached here. In this case, those are 17 datasets. The IDs here are rather cryptic, but we can, for instance, toggle the view here and show a metadata overview. And here we get all the covariates that are later important for our analysis, for instance, age, sex, the batch, or disease group. And if we select an individual data set and move down to the detail view, we again get a list of all the metadata, but we also have this ProData tab here that lists all the processed data sets that are associated with this data set. And this is in this case, for instance, a count matrix in H5 format, which is the input for our single-cell RNA-Seq analysis. And if you want to learn more about this ProData feature, I'm going to link a video here where you can see how you can upload and attach ProData for individual data sets. So we are now in Visual Studio Code, where I'm connecting to my Jupyter server. And I'm going to walk you through the individual steps here that we run for our single-cell RNA-Seq analysis. So we begin by performing the imports here. We need PyReadStore. This is the Python ReadStore SDK. And of course, Scanpy, Pandas, and then AnnData, which is the data format for Scanpy. So Scanpy users know this. And then the first thing we're going to do is create a ReadStore client, and we need the client to access our credentials to connect to our ReadStore server. I'm going to link the video here if you want to know how to set up and configure your client in a way that you can access your user credentials to connect to the database later. Once we have this client here, we can start to get a list of all the projects that we have available. If we run this here, the output is already a Pandas data frame, and then we see that we have here our SAVI study, including the metadata and the attachments and the project ID. And the first thing we're going to do now is access all the is access all the data sets now for our SAVI study that we need later on. And here we can use the ReadStore client list command, and we will provide the project name that will pull exactly those data sets out that are part of this project, and we don't need to search or filter anything anymore later. List of data sets contains different variables, the ID and the name, and then of course information on the project and the metadata as key value store. And we are going to convert this now, this dictionaries here for the metadata, into a separate data frame. This is what is happening here in this line. We're just casting this into a series and then attach the metadata with the dataset name. And this gives us a nice data frame with all our covariables and the dataset name here. In the end, very helpful because we don't need to import any metadata from Excel files or any other cryptic format. We have everything in a very clean way and can access it within a few seconds actually. And after we configured our metadata, what we're doing in the next step here is get our input files for the single-cell RNA-Seq analysis. And those are stored as pro data, as processed data sets, because they're not the raw FASTQ files, but they've been processed by Cell Ranger pipeline, for instance, and we've stored these count matrices in our ReadStore database. And we can access those again with this list pro data command, where we filter again for the project and for the data type. And the data type here, we've seen this before, is the count matrix in H5 format. And this will exactly give us the path to all the processed data sets or single cell data, and we'll see this here. If we scroll down here, then we have now the count matrices, and here is the path to all of these files, and we can load our data sets from this path. There's no more searching where exactly the files are. It's all available through the database. Next, we are going to loop over the dataset names and the path in order to load those using Scanpy, and we use here this read 10x h5 command. This is going to create an AnnData object, and we are running this unique variable names command. And then we want to attach the metadata that we just loaded for each of the cells that are part of our AnnData object. And for this, we subset here the metadata for our sample, and then we need to perform this transformation here. So we have to cast the metadata. I mean, this is a one-dimensional vector. We need to cast this now to match it to the shape for all the cells in the AnnData object. People that want to learn more about this can leave a comment, and I can explain this in more detail, but this leaves us with these ops object here, which will attach to the AnnData object here. And this will basically set for each of the cells in the AnnData objects the respective metadata, so we have everything nicely annotated for our downstream analysis. And finally, we are going to set this as part of a dictionary. And this dictionary we can then later use to concatenate all those 17 individual data sets. But before we do this, what we want to do now is clean these AnnData objects and again save them in our ReadStore database, so that the next time we don't actually need to load the H5 formatted feature barcode count matrices, but we can actually directly load the filtered AnnData objects. And here we are running now in a list comprehension. We are going to run now this Scanpy filter cells and filter genes operations for each of the data sets. And this is now filtering out all cells that have less than 100 genes detected and where genes are detected in less than 3 cells. And this is cleaning a large proportion of our data sets and also leaves a smaller footprint of our data. And in the next step, we want to save those cleaned AnnData objects. For this, we prepare here a file prefix where we want to store the files. And then we loop through this dictionary here and use the AnnData write command to write these to write each AnnData entry to a separate file. And most important step here, we want to also upload and register these files now in our ReadStore database. And for this, we use the upload ProData command, where we need to give the process data a name. We have to give it a data type, which we're going to set now here to AnnData. Again, we can then later filter for this data type. As metadata, we attach now here the filter criteria here for our data sets. And of course, we have to provide the name where the file is stored and the dataset name that this process dataset is going to be attached to. And if we run this command, we create the respective entries in our ReadStore database. And we can again check if this has been successful. And if we run this list pro data command here, we see that we get an overview here of all the process data sets that we have just uploaded. And the data type here is now AnnData, and we get a path to those files. After we've prepared our data sets and saved the processed AnnData into our ReadStore database, now we're going to the more single cell analysis, and we start by concatenating all our AnnData sets into a single vector, because we want to have one object that contains all the cells from the 17 different data sets. For this, we use this concat operation, and this will also maintain the metadata that we stored for each cell. Then next, there are a few standard steps. I think there are a lot of great tutorials for single-cell RNA-Seq, where you can have a look what these individual steps do and how you can adapt them. I'm just quickly going through those. So we are normalizing the cell counts here and performing a log transformation of our gene counts. And then there are three standard steps for single-cell RNA-Seq analysis. So first we look at the genes which are most variable between our cells that help us to later on perform the clustering. We perform a neighborhood calculation, so we check how cells behave to other cells, and then finally we calculate UMAP embedding, so dimensionality reduction in order to visualize the cells later. And I think the people that work with single-cell genomics know these plots very well. And after performing this, we have done we can now plot the data sets and make use of the metadata which we attached in order to visualize the cells. So for instance, here this UMAP, we highlight the sample IDs here in the legend. In the next example, we highlight the batch information that we stored for each of the data sets in our samples. And finally, we have the disease group here. And with this type of well-annotated data frames, it's now very easy to perform a routine downstream analysis. And ReadStore helps you with its SDK interface to fully standardize these analysis workflows, because you can always go through the same types of transformations of your data. And if you now have another project, for instance, where you have other types of metadata or different numbers of samples, you could automate these scripts, or you could automate your bioinformatics workflows to every time go through the ReadStore database, pull the relevant data, and you don't have to manage any metadata anymore by hand or with Excel tables that are very error prone. So I hope I could motivate you to try ReadStore. There's a demo available on our website for you to try out. And if you have questions or suggestions, please reach out, leave a comment. I'm happy to answer any questions. And with this, I wish you a great day.